All right, so this is our first lecture for statistical analysis part of CAP biology. This might not seem like biology, however, being able to test hypotheses with well thought out experiments is science. So, the reason why we're doing this as our first unit is to get a lot of background for different labs and things like that we're going to be doing for the rest of the year. So, we know that within any population, there's going to be variation. And within that variation, there is certain variability. What that means is in a whole population, that is the number of things that are different and how different they are. And that is going to affect how confident we are in our conclusions. What that means is if we say, all dogs are about 40 pounds. Well, within the dogs that we have, there's huge variability. Some dogs are 3 pounds, like a little Chihuahua, and some dogs can be over 200 pounds, like a Great Dane or a Mastiff. So we have to look at that variability to get confidence in that thing that we said that all dogs are about 40 pounds. So we're looking at the whole variability, we're averaging together, doing some statistics to try to figure that stuff out to see how reliable we are. On a less funny note, not like the dog example, this is used quite often in medicine. So, when you have to buy your own medicines for certain things, we have generic medicines and then proprietary medicines. Those are like the name brands. So, for example, a name brand medicine that we have is Tylenol, where the generic for that is going to be acetaminophen. Okay, so which one do you choose? Would you buy a store brand acetaminophen, or are you going to go with a proprietary Tylenol blend? If we look down at this chart, it looks like that proprietary blend is way more effective than the generic. However, now when we add in these lines, so these lines are confidence intervals or error bars, okay? And we see they have a ton of overlap. So before, in the previous slide, where it really, really looked like the proprietary was way better than the generic. Now, when we're looking at these error bars, we can actually see that they are pretty similar in how effective they actually are. So, why do we need the statistical analysis? Well, we need it to evaluate the data that we have, to make sense of all the numbers. And this is where we get to what is a hypothesis versus what is a prediction. So if we look at all of these statements on here, which one are actually scientific hypotheses? So if we look at the first one, if fertilizer is added to soil, then the plant will grow taller. So typically we learned before if we have an if then statement, that is a hypothesis. So let's look at the next one. The deformities were caused by one or more chemicals in the water. That is a statement. If I add salt to fresh water, then the water will freeze at a lower temperature. The farther something falls, the greater speed it attains. That seems to be a statement. Hypothesize what will happen to the pendulum's motion and final height if swing is interrupted by the cross arm. Well, that seems to be a question. So, which one of these are hypotheses? Well, you probably learned in a science class that number 1 and number 3 were hypotheses. However, it's number 2 and number 4. A hypothesis in science is a statement that says, I know what is going to happen, and then I'm going to try to prove that. So, in cat biology, when we are looking at these hypotheses, statements like number 2 and number 4 are going to be our hypotheses. So, deformities were caused by one or more chemicals in the water. Now, I need to try to figure out what those chemicals are. Or number 4, the farther something falls, the greater speed it attains. Now I have to try to prove that thing. So, like I said before, a lot of science we kind of think about as an if then statement. However, I can turn that into a hypothesis. We're not really predicting anything, we are explaining an observation. When we are looking at these hypotheses, we are going to focus on this last one down here, the null hypothesis. This is going to be the hypothesis we're going to use for everything in science. So, trying to explain and trying to generalize, we are going to cross out and we are going to focus on this guy down here, the null hypothesis. This is going to be the big key point. What this null hypothesis is saying is that there is no significant difference between specified populations, and any observed difference is used to sampling or experimental error. This is going to make more sense in a little bit. Okay? So, if we are looking at these hypotheses, an explanatory hypothesis would say the speed of an object depends on the air resistance and the size. That is explaining what is happening. A generalizing hypothesis is making a general statement about the way the world works. So, the farther something falls, the greater speed it attains. A null hypothesis seems to contradict both of these things. So it is saying there is no significant difference between the height of the fall and the speed the object attains. What this means is having a null hypothesis is that we can always prove this right or we can prove it wrong. And the reason why we are using a statement like this is because it's easy mathematically for us to figure out a null hypothesis. So, for an example of this, I have a little example with birds. So, if we have hummingbirds, they are a nectarivore, so that means they only feed on nectar. And in return for food, they pollinate the flowers, so it's a mutualistic benefit. So, as a result of natural selection, the hummingbird's bills, these long guys right here, have evolved along with our flower to be this really, really unique shape. So, if we are researching 2 different species of hummingbirds, we're going to use some comparative anatomy to collect data on bill length in 2 species. So, we have a red-throated and a broad-billed hummingbird. So, in order to do this, we need to see if there's a difference between the lengths of these bills, and then maybe we could figure out what flowers that these hummingbirds will exclusively feed on. So, we are going to need to do a null hypothesis. So, what that null hypothesis is saying is that there is no significant difference in bill length between the 2 species. The sample size has to be large enough for us to get sufficient data to do the statistical testing. And we have to be mindful of uncertainty and error. So, when we're testing animals, if we recatch the same animal and measure it over and over and over again, we're not going to get reliable results. So, we have to mark our samples, maybe collect at the same time of the day, different locations, anything like that. So, just for some examples of null hypothesis, I just literally typed this into Google for some examples. So, with our little null hypothesis down here, if I wanted to prove if cats liked one food or another, I would say cats show no preference for food based on shape. So, in my experiments, I would do an experiment to try to prove that they did like the different shapes, but my null hypothesis is going to say there is no preference. So, in my next experiment, if I'm experimenting between 2 different plants and what different colors of light will do in the growth, my null hypothesis is going to be plant growth is not affected by light color. Sorry, my baby's crying. And then for the last one, if I want to see if age is indicative of ability of people to play musical instruments, I would say the age has no effect on musical ability. And then in all of my experiments, I would prove maybe otherwise.